glow as a function of some activities that they performed. So it's pretty cool within that respect. The lab itself, the kit is turnkey, so most of the materials that you'll need to prep as well as implement the lab in and of itself are all included in the box, just a few supplemental activities for preparation. It accommodates a great deal of students, or a great volume of students, up to 32 students, wherein we make suggestions for 8 groups with a maximum of accommodating 4 students per group. And we know that lab techs as well as teachers alike don't have a lot of time. So it's great in that it's an investigation that can be implemented in generally periods, depending a third, depending on if you want to embrace the final extension activity. And that's within a 45 to 50-minute class session. So it's pretty cool and accommodating with that respect. All right, so general sequence of student activities. Within the laboratory investigation, day 1, students perform a genetic transformation. That is, they're going to transform cells and spread it across their plates and allow it to incubate over 24 hours or more. We'll talk about those accommodations or adaptations a little bit later. Day 2, they absorb their transformants and controls. They analyze and interpret their results. And in day 3, optional as an extensional activity, they can perform or calculate transformation efficiency of their transformants, which a lot of teachers tend to embrace to kind of give them, you know, a mathematical side or view of their scientific outputs. All right, so a little bit of background so that you can better understand the context of the lab. The overarching goal here is, again, to perform a genetic transformation. We are going to move the ring fluorescent protein from the Aquaria Victoria organism, which is a eukaryotic organism, common jellyfish, over to prokaryotic organism, in this case, Escherichia coli HB101K12 strain, which is a safe laboratory strain that's been used for many of years. So no worries about students being harmed, although we do recommend that you oblige all safety and best practices when performing a microbiology experiment here. All right, so in order to achieve this, again, students will be performing a genetic transformation. And so as a reminder, a genetic transformation is just a process that involves the introduction and expression of a foreign gene, in this case, the GFP gene, over to a host organism, which in this case will be the E. coli. In order to accomplish this, we're simply capitalizing on processes that bacteria have used for millions of years, and that's sharing genetic or swapping genetic information between one another by way of plasmids. As a reminder, bacteria often have plasmids, which are extra chromosomal circular loops of DNA that often code for proteins that confer some sort of benefit or advantage in an environment, such as antibiotic resistance. Right, bacteria can take up new plasmids, and again, we can capitalize on this because scientists can modify or engineer plasmids for specific purposes. So in this case, to produce or for proof of concept that a foreign or host can take in a gene from another organism, even cross species or in different kingdoms, and express a gene as long as they have the cellular wherewithal, which is pretty cool in this case, again, because the gene of interest here is a gene that actually glows under long wave UV light. All right, so our construct, what students will be working with, is something called the PGLO plasmid, right? Named after the gene of interest highlighted here in green. But it's important that you understand that some of the constructs here are put in place for very specific purposes, and we'll go into further detail here in the succeeding slides. So we have an ORI, and that actually helps with propagation of the plasmid in successive generations. Of course, our gene of interest, we have a way of providing protection under a selective pressure with this particular gene. An RSC gene and PBAC promoter that helps to regulate the expression of GFP so that it's not constitutively expressing. All right, so if you can see, I'm highlighting areas of our PGLO plasmid in the left. on the screen. And so we're going to emphasize the role of a particular construct within the plasmid on the screens as we move from slide to slide. So again, the ori, which is the origin of replication, assures propagation and duplication of, or duplication and propagation of the plasmid that's successfully uptaken by any of the bacteria, Okay, and so before moving on, just saw a comment that someone cannot see the slides. I don't know, Gary, if you can accommodate that individual, or if this is a problem being experienced by a good deal of us at this point.
Yeah, can a few people message in if they can see the slides? Okay, someone's already messaged saying they can. So again, I think it's going to be, yeah, everyone else can, it seems. I'm not too sure why you can't see the slides. Oh, sorry, I've had plenty of people messaging now. So thank you for the feedback.
Thank you so very much, everyone. Greatly appreciate it. All right. So again, this area or this part of the PGLO plasmid is going to assure that as the bacteria that have been transformed successfully are able to, as they're dividing, to copy the plasmid into successive generations, so that they too will then have the conferred properties of the ampicillin resistance, what we'll talk about here in a second, as well as are able to express the gene of interest. All right, so the next area or next part of this particular plasmid is the ampicillin resistant gene. All right, and specifically, this is going to produce beta-lactamase. So this is a gene for beta-lactamase, and it is highlighted here in a couple of the bacteria on this screen in red. Now, beta-lactamase, when expressed, will produce a protein represented here in red, the enzyme that is responsible for degrading ampicillin. Now, this is going to help the bacteria who are successfully transformed to survive in an environment that contains ampicillin, which is part of the prep and as part of the experience for students, so that they can validate successful transformation, or this being one of the ways that they can validate successful transformation of their bacteria. Right? So again, if you successfully taken up the plasmid, you will survive in an environment of ampicillin. Those who did not will not. All right, and then of course there's our gene of interest here, okay? The wow factor of this particular experience, all right? And that's the green fluorescent protein. And so just to build a little bit of familiarity with this particular protein, it is a tertiary structure. It confers a barrel structure with beta pleated sheets that surround a central alpha helix. The alpha helix itself contains a chromophore that's excited at a wavelength of 395 nanometers and emits at a wavelength of 509 nanometers. Therefore, there is a UV pen or UV wands that can be purchased separate from the kit that are utilized to illuminate or to excite the chromophore, and that allows students to visualize in a very, very glorious way whether their experiment or the transformation was not successful as seen here under UV light, or if it was successful as seen under the UV light here, which is something that always produces the wow factor. So students get very, very excited about this. And just a side note story, again, these are very safe, a very safe strain of bacteria. It was shared with us in one of our experiences in training teachers on this, that the students were so excited about the glowing bacteria that they wanted to to see if in fact they could process that particular protein, and if it would kind of come out on the other end. So there was a student, believe it or not, who licked the successful transformed plate and took the UV wand home to see if it would appear in their excreta. So that was an interesting story to tell. One, a testament to how safe it actually is, because the student was just fine. But two, adventurous students, and just taking their level of excitement from the products of this particular lab and trying to take it at another level. So very interesting stuff. All right. Also within the construct, we built in ways to regulate the GFP gene, because with all organisms, we don't want every gene on at all times, simply because it's a waste of energy, especially if you don't need that gene at a particular moment. All right, and so to better understand how we regulate the expression of the GFP gene that students will visualize in the end, we need to understand a couple of the constructs in place, and that's the ARC gene as well as the PBAP promoter. Now, this area that you actually see to the right of the screen, these constructs are representative of a modified Arabinose operon. So again, to understand this mechanism a little bit better, we'll just kind of review very quickly the Arabinose operon. All right, so just as a reminder, an operon is just a cluster of genes under control of a single promoter. Okay, and the goal of an operon is to express structural proteins. In this case, seen in various blue shades, Arabinose B, A, and D. Thus, the name of the promoter, PBAD, which is representative of the structural genes that it actually controls. ARC, there's a dimer here, and that is produced from the ARC gene that you actually see here. All right, and that part is actually constitutively, expressed, dimerized, and that controls whether RNA polymerase has access to the promoter area, which then allows active transcription. So the only time this is permitted is when Arabinose is present in the environment. And the goal of that is that when glucose levels are low and bacteria need another carbon source for energy, Arabinose can be readily used by turning on or activating this particular operon, which then transcribes create a protein, which leads to an enzyme that then can catabolize Arabinose and then allow them to access energy for it. Now, and again, as I indicated, we wanted to better understand how GFP is being controlled, so we took a look at the Arabinose operon. So what we have done, or what's included in the plasmid, is a modified version of the Arabinose operon. Notice that the ARC gene still exists, the PBAP promoter still exists. However, we swapped out here the structural components with the eukaryotic gene from the jellyfish, the GFP gene. So by way of the same mechanism, we will control the expression of GFP, or we can control the expression of GFP by that very ARC dimer, which is again constitutively expressed, that actively represses the transcription of this particular gene, unless we provide Arabinose in the environment, which will then bind to the dimer, change the configuration or conformation of that particular protein, and then allows access of RNA polymerase to then actively transcribe GFP. And so note, one of the common questions that we often get about the structure of the PGLO plasmid is, why are there arrows in two different opposite directions? So I'd like to bring your attention to the ARC component of the plasmid here, so that you know, in order to be expressed constitutively and separate from the GFP and the other genes here, Arabinose is under its own promoter, which is located adjacent to the PBAP promoter, but it runs in the opposite direction. So that is why we have representation here of bidirectional arrows. So again, what's not seen here is the promoter ARC, which again needs to be on all the time so that it can actively repress the expression of this unless Arabinose is in the general environment. All right, so as a review, students will be chemically inducing the uptake of this PGLO plasmid into bacteria, E. coli in particular. And with that, it will bring along with it the ability to replicate the plasmid, bring along or serve as a vector for the gene of interest, GFP in this case, provide some sort of ability to thrive in an environment with ampicillin, so to outlive a selection or negative selection factor. All right, a way of controlling the expression of GFP. All right, so before we jump into the lab prep and student station setup, are there any looming questions about the background?
I'm not seeing any come in the chat yet, but remember, if you do want to ask a question, if you raise your hand using the hand icon at the top, I can unmute your microphone and you can ask a question. But I can't see any hands raised at the moment, Tamika, so I think we're okay to carry on.
Fantastic. Okay, so as it pertains mostly to the group represented here in this webinar, we're going to go into the specifics of lab prep as well as student prep. So again, I say that this is a turnkey kit. So a lot of what you need is actually in the kit. A couple supplemental things like distilled water, and then of course glassware, some sort of heating source, which we'll get into here in a moment, is all you'll actually need. Now, when you receive your kit, storage is very easy. All you simply need to do is the refill, the transformation reagent refill baggie that looks like this. If everyone can see this. Simply toss that into the refrigerator until you're ready to actually prep. The remaining materials can remain in the box at room temperature. All right, so here are a list of the kit components here. What I'd like to draw your attention to, in addition to all the other items provided, are the lyophilized components. So you have four different, you have 4 items that are lyophilized, and obviously, and that's for storage, to increase the longevity of storage, which then means, of course, you need to reconstitute it as a part of your preparation activities within this lab. And that includes the bacterial specimen, the PGLO plasmid, the ampicillin, and the Arabinose. All right, so some of the required materials, as you might anticipate, some sort of heating source, microwave, you may autoclave to create your auger plates, and so that's different depending on the institution. Obviously, ways to monitor the temperature of your auger, distilled water, etc., and recommended materials to acutely monitor volumes of the added liquids, as well as be able to specifically incubate. To have an incubator oven, vortex for a vortexer for a reason that I'll raise here in just a second, etc. So not a lot of items necessarily needed in order to prep this particular lab.
We've had a question come in from Helen, Tamika, asking how long do the materials last in storage? If they bought a kit, when would they need to really be doing it?
Excellent question. So we guarantee that it lasts up to a year.
Okay, plenty of time. Lovely.
If properly stored. Very good question. Thank you. All right, so your preparation timeline can be seen in 3 defined clusters. Okay, you'll start your prep 3 to 7 days prior to students actually implementing the lab. And so 3 to 7 days prior, this is where you're going to pour your plates. You're pouring 3 different types of plates, including LB, which is an enriched medium for the bacteria to grow. You're pouring LB Amp plates, which are the LB medium, including the ampicillin for that selection pressure. And you'll also be preparing LB ampicillin Arabinose plates. And again, that's 3 to 7 days prior. The further out that you prepare your plates, obviously you store them in a cool environment, refrigerator, until students will be utilizing them for their lab. 24 to 48 hours, you will be streaking student starter plates, and you'll be producing 8 of these and aliquoting students' materials or reagents for the lab setup. And then immediately prior, you're going to sequester and organize student station lab materials and place it out for them to execute the lab. All right, so particularly focusing on what's happening potentially up to a week before the lab, you're going to be preparing auger plates. Now, the kit materials required for this are Arabinose, ampicillin. You'll be utilizing a transformation solution, or you could use sterilized water, although a transformation solution is a convenient alternative to rehydrate or reconstitute your lyophilized components. LB auger, the 40 Petri dishes, and additional items required for that will be a flask, 500 milliliters of distilled water. You could use a P20 micropipette, and or again, kind of staying in line or within the vein of the turnkey type of experience, you can utilize the sterile plastic transfer pipettes that are provided in the kit to aliquot or to transfer accurate volumes in preparation. And then, of course, some sort of heat source and thermometer. We anticipate or we recommend that you allot maybe 1 to 2 hours to prepare the auger plates. All right, so at this point, we're going to view a roughly 4-minute video that comes from our arsenal of resources. So this gives you an opportunity to see some examples from our instructional supports and even lab prep supports, and have an opportunity simultaneously to watch how you will pour these 3 distinct plates. Now I'll pause here, okay.
I could hear the video.
Oh.
I'm not sure if I assume most people could hear the video. If you can't hear, I think it's more likely to be, again, your school speakers. Maybe if you have a headset, try plugging that in instead, possibly.
Okay, and so alternatively to the labeling at the bottom of the plate, we do recommend sometimes just for easier labeling, and let's see if we can all see this, that you label your plates or just make by creating markings on the side. And so one stripe or a black stripe, which is would then represent LB. You would then mark the LB amp plates with a black and a red stripe. And then finally, you would mark the LB amp Arabinose plates with 3 stripes, black, red, and green. Okay, and so I wanted to present that as an alternative or shorthand way of labeling plates as well. So hopefully most of us had an opportunity to see and experience that. I did see a couple of comments pop up as the video was playing. All right, so a couple of key technical reminders to this part of the prepping process to keep in mind are that best results are obtained when prepared in a flask. Again, one that is 2 times the volume of the auger solution that you'll be preparing, which is a total of 500 milliliters. So you want a 1 liter flask. Allow the flask to cool before swirling, particularly if you are heating your auger contents in a microwave to avoid a flash boiling event. Again, as indicated earlier, sterile water can be used, but transformation solution is provided and is a convenient option to reconstitute your lyophilized materials. Please note that the Arabinose does take time to dissolve. My experience essentially has been about 15 minutes when it's kind of stationary, so 15 to 20 minutes if in fact you add the transformation solution and let it alone. But vortexing does actually help to break it up, increase surface area, and then allow it to dissolve a little bit faster. Keep in mind that temperatures above 6 degrees Celsius can degrade the ampicillin and Arabinose. So you want to make sure that you balance that, or make sure that you have general awareness of that before adding those 2 constituents. All as being keenly aware that auger solidifies at 27 degrees Celsius so that you don't have a solidifying event before you have an opportunity to introduce those reagents to pour those specific type of plates. And that, again, plates can be stored ideally up to 4 weeks, although we recommend for optimal experience to do this maybe a week out. So as indicated, 3 to 7 days prior to students running the lab. However, if your plates are sealed well, they can last up to about 4 weeks, but we recommend no longer than 4 weeks to utilize it for this experience, or for an optimal experience at least. All right, next you'll be, the next stage of your preparation process is dedicated to streaking and storing your starter plates. And this occurs 24 to 48 hours prior to the student lab or activity. All right, so this constitutes transferring Laurier broth, or the LP broth, over to the lyophilized E. coli, 250 microliters in particular. This then needs to be incubated at 37 degrees Celsius from 8 to 24 hours, or it can be incubated at room temperature, but it will require a little bit more time, so 16 to 24 hours before you can actually streak your starter plates. And you want to make certain that you're storing your LB at 4 degrees Celsius until use, simply because if there was potential contamination, we want to retard or mitigate any type of growth within the broth before it's actually utilized. So after you reconstitute this and the bacteria have had an opportunity to incubate, you're going to streak starter plates. We do have another video dedicated to showing you how to actually streak your starter plates. But the general pattern is shown here, illustrated here, to wherein you want to maximize on as much surface area of the plate as possible, being careful not to gouge the agar and to just gently swipe. So what that constitutes or looks like, is that you will simply dip your inoculating loop that's provided with the kit, and then obtain essentially 10 microliters of the culture here, and that actually should look, I don't know how well you can see that, but it should look like a bubble, a small film within the loop. And that's all you will utilize for each plate. So you dip once, and then you swipe back and forth in one particular quadrant very lightly, and then you will turn your plate 45 degrees, and then you will streak back and forth, making sure to include or to go over some of the streaks created in the previous streaks, so that you transfer yet dilute the concentration of the bacteria within the second set of streaks, and continue that and rotate it until the entire plate is streaked. The ultimate goal for this is to assure that you have active colonies growing so that the material is diluted enough so that you can have active colonies growing, because the active colonies perform best when performing the transformation. Right, and so you will make 8 starter plates and incubate this, and incubate the starter plates at 37 degrees overnight, and they should be ready by the next morning. So time according to that, depending on if you have an incubator. But if you are without an incubator and you need to incubate at room temperature, provide at least 2 to 3 days to get those active colonies growing or visible. All right. You're not to use the plates after 36 hours, because this is when it has reached a stationary phase or death phase, and so you're not getting as active of cells in order to perform or to optimize the transformation experience.
Just to interrupt for a second, we've had a question about what happens if you incubate bacteria for too long. If you left it in the incubator way longer than you should, do you know what happens to the bacteria?
So yeah, they'll enter the stationary phase. They only have short lifespans. So again, the idea here is to make sure that they are actively growing. But again, they will begin to reach a death phase. So the stationary phase, when we're looking at bacterial growth, the lag, the log, the stationary, right, and then the death phase. So they'll enter that stationary phase after a while, as well as the death phase, and then they will no longer be alive. It cannot be used for the experience.
Lovely, thank you.
You're welcome. All right, so alongside of creating those, prepping your starter plates, you'll also a la carte reagents for students, which includes each student being provided with 1 milliliter of transformation solution. We provide color tubes for this, and so the blue tubes are dedicated to the transformation solution. You'll also label the tubes accordingly so that students are aware of the contents or have some sort of color code associated with it as well, if that's the secondary way of relaying the contents of the microtubes. Okay, you'll create 8 setups of this. You'll also transfer 1 milliliter of the laurea broth to the yellow tubes. For students, you'll repeat this 8 times, right? And then you will reconstitute the plasmid. Now, this won't be aliquoted for students. Rather, this will be shared among students. And so teachers have different ways of doing this, but for your particular role, your goal is to make sure you just simply reconstitute it and provide the vial to the teacher so that they can make the judgment or make the call on how they want to share the plasmid with students during the general experience. All right, so once everything is aliquoted, you will create 8 student workstations to include the materials listed here, including a starter plate, 4 plates, 4 of the poured auger plates, including 1 LB, 2 LB amp plates, and 1 LB amp Arabinose plate, 2 with this transformation solution, LB broth, a package of inoculation loops, pipettes, 5 of those. You'll include a foam microtube holder, and that comes with the kit and looks an awful lot like this. And so you just kind of separate those out and provide one with each student stationed. Containers for trash, 1 with crushed ice for the transformation, a marking pen, and additional microcentrifuge tubes to perform the transformation in. And then within a common workstation, you like to set up a water bath for the heat shocking or the transformation process, which is part of the transformation process, a UV light that's going to be shared as well. And so that all goes into a centralized area, as well as an incubator and additional materials such as micropipettes if desired.
Questions come in. What is the purpose of ampicillin in bacterial transformation?
Okay, so it's going to be a way of selecting against any, let's say, contaminants, right? And to assure that the bacteria, or one way to verify that the bacteria have been successfully transformed. So again, on that PGLO plasmid, one of the areas was the ampicillin resistant gene, which codes for beta lactamase. and that beta-lactamase is going to be an enzyme, or is an enzyme, that will degrade the ampicillin so that it is able to thrive within a plate that contains ampicillin, whereas other organisms who do not have the PGLO plasmid will not be able to thrive in an area or a plate that includes ampicillin.
Thank you.
Okay. All right, so some social distancing adaptations here in the event that this is still a real thing for labs in your area. We've developed out a way where you can create 24 individual student stations. This is all nicely outlined in our social distancing adaptations, modifications resource, and that can be found on our website. But I wanted to kind of give you a preview of what that looked like, and so how we break it out in a visual manner, as well as how we write it out so that you understand how to modify the aliquoting of solutions, as well as how do you set up this particular lab that's generally set up for 8 student groups to accommodate 24 student individual lab stations. All right, so the student-facing part of the investigative protocol. What will students see during this investigation, and what will they learn, essentially? So students have the option, or you guys have the option, of providing students with a digital micropipette, or allowing them to use the disposable sterile transfer pipettes that are provided in the kit. What they will need to be aware of are the volumes, the volumes that are that equate to specific volumes on the gradation points. More specifically, they'll need to be aware of where how to measure out 100 microliters, 250 microliters, and 1 milliliter. Now, if you were utilizing this for prep, you would want it to be aware of the 500 microliter point, which is the midpoint gradation point on this pipette. All right, so again, day 1, students will be performing a bacterial transformation. All right, the lab looks like this. Very simply, students take the extra microtubes, they label it plus and minus PGLO. They'll then transfer 250 microliters of the transformation solution over into both tubes. Now, the goal and the purpose of the transformation solution is to provide a source of calcium, and the calcium is going to help to shield the negative charges of the DNA that's probably or being introduced to the bacteria. Now, recall that the membrane in and of itself has an overall negative charge, which then creates these repulsive forces. And so in order to get or bring the plasmid closer to the membranes, so that once we perform the heat shock step, that it is likely to come into or come inside of or be uptaken by the bacteria, we need to negate or neutralize the charge of the DNA. And so the transformation solution assists with that or facilitates that. We then place both of the tubes on ice. Then we'll introduce colonies of these starter plates. And so I don't know how well you're able to see this. This is an example of starter plate. So we would want the individual colonies in there. Let's see, get it out of a blurry view. So you may be able to see some of those dots in there. And so you want students to pick off those particular colonies. Those are actively growing. We know that they are a pure strain, and so they've all descended from one initial bacterial cell, and then so that's what generally a colony actually is. So you want the colony to be about 1 millimeter to 1.5 millimeter in diameter, and you want students to transfer over 2 to 4 of these into each tube using the inoculating loop. Now, I know that most students will see this and say, well, more is better. And that's not the case here. So you want to make sure that students aren't transferring more, because that then does mitigate or minimize by tenfold the number of transformants that are, or well, the number of transformants that come out of this. So again, you're hampering the process by introducing more because they're competing for resources and what have you at that point. All right, after the colonies are transferred into the tubes, the students will place it on ice, and that's for a purpose we'll look at here in just a second. The students will then introduce a loop full of PGLO plasmid. This is where that vial comes into play, that single vial where the teacher can pass it around, or again, it's up to the teacher how, the instructor, how they want to aliquot this. This can be by way of a digital pipette, or you can use the inoculating loop, which the loop itself, once it contains a film or is dipped, it contains a film, roughly has about 10 microliters of the PGLO plasmid, so they can dip and then transfer just specifically into the tube labeled plus PGLO. All right, then they will incubate their tubes on ice for about 10 minutes, and then they will perform the heat shock process. Now, it's very important that students follow the instructions seen here because this is an optimized protocol. Again, if you deviate with the temperature in terms of the heat shock, one way or the other, you can decrease the number of transformants, tenfold or by half, depending on which direction you actually take the temperature. So you'd like to set your temperature to 42 degrees Celsius, and you want to expose the tubes after coming off of ice after 10 minutes for just 50 seconds, and then reintroduce it to ice for 2 minutes. Now, the goal of this is to create pores in the cell membrane, right? So that's the heat shock element. So again, we've used a transformation solution to neutralize the charge of the plasmid DNA, get a little bit closer to the cell membrane of the E. coli. Putting it on ice helps to immobilize some of that fluid movement of the tails of the membrane, and so helping with the mobilization helps to create a clean break and thus a nice clean pore once you actually introduce it to heat. Allowing the plasmid to drop in, and then we obviously want to drop the temperature very quickly to close back up the pore and to seal in the PGLO plasmid inside of the bacteria specimen. Thereafter, after 2 minutes having it on ice, you want to bring your tubes. The students will bring their tubes to room temperature. Add LB broth to help with recovery, flick it so that the bacteria within the tubes have access to all of the nutrients made available by LB. And they're going to incubate at room temperature for 10 minutes. This is the lower threshold in which they could actually incubate, but it can go a lot longer. The goal being in this recovery period, permitting the E. coli an opportunity to access some of those nutrient resources and then to begin to produce some or express some of the genes that are included in the PGLO plasmid, so that it is actually ready to be transferred over to the plate. Now note that the recovery culture can incubate at room temperature up to 24 hours. Therefore, this is a good stopping point in the lab, and thus you probably wouldn't want to have the plates ready for students if in fact it is the option of the instructor to stop at this point and incubate at room temperature for 24 hours. All right, again, students will be provided with label plates. They will then add their initial, their group's name, however they want to go about labeling it. They will then introduce 100 microliters of contents from one of the tubes, and then they will use the inoculating loop to spread the bacteria across the plate, close it up. All right, seal it, and then they'll invert it. Now, very specifically, they will be adding the contents from the plus PGLO plasmid tube to the LB Amp plate, the LB Amp, and then the LB Amp Arabinose plate, and then contents from the negative plasmid tube to the LB Amp and LB, which are your controls, and your experimental groups are your LB Amp and LB Amp Arabinose plates. Again, these plates will then be stacked, sealed or taped together, inverted, and incubated overnight at 37 degrees Celsius. Any lingering questions about the student-facing part of the lab, or at least the bacterial transformation?
I was on mute, but yes, we did have a question come in, which me and Don are not sure the answer to. Does it work with 1 microliter of plasmid?
The success rate is not as high. We're sure to get some transformants, but you won't. And that's the question that's being asked. Can you use less plasmid? You can, but again, the transformation rate won't be as high, and so you may not see as many successful glowing colonies in the end.
Right. Okay. Thank you. Oh, can you use? Yeah, can you use less plasmid? The same question.
Yep. Great questions, guys. All right. So results in analysis. What will students actually see? So I don't know how we would go about it with so many participants here, but I thought that we do a little bit of predicting here to see, again, how well we've been able to internalize some of the information, because it's been kind of a fast and furious introduction to this information. But I have the conditions listed below, not respective to the plate that's being demonstrated here, and see if anyone want to kind of give it a go if they'd like to match the condition or with the anticipated results. And again, this is all viewed under UV light. No one? Okay, I realize there are quite a few of us.
Yeah,
All right.
I was worried we get tons of messages of all answers coming.
Okay. Okay, the glowing one is, okay, so we do have some answers coming in. The glowing one is PGLO Plus with ERA.
Any other guesses or?
Okay, so thank you for that contribution there. And here are the answers for the alignments. So the plate that you actually see with colonies, but that are not glowing, obviously, is that we don't have the Arabinose there to regulate the transcription of the GFP gene. However, we have the bacteria have successfully taken up the plasmid, as they are still able to grow in the presence of ampicillin. And we had a participant who identified correctly. The glowing one is the one with the PGLO plasmid and is able to kind of survive under the presence of ampicillin and have the sugars to regulate or turn on the GFP gene on the plasmids. You have minus PGLO on LB amp. Obviously, if you are plated, or if the bacteria are plated in the presence of ampicillin without the plasmid, they're not going to be able to grow. And then without some controlled conditions, such as having ampicillin and Arabinose on a plate, that the bacteria will grow without the plasmid, but they will grow analon because their growth is unchecked. All right, again, in the end, and this is optional, students can perform transformation efficiency. Steps to performing this are outlined in the lab manual. So if you want to know more specifics about this, the information can be outlined there, or is outlined there.
Should you not also have a PGLO Amp Arabinose control? That would just be a blank Petri dish, wouldn't it? A control one?
Correct.
Yeah, I don't think it's necessary for the experiment.
Correct. But I understand the reasoning for the question, because we get the question all the time.
Mm-hmm.
All right, so we have a slew of prep resources. Again, within the Bio Rad Explorer program, you had an opportunity to view, or at least most of us had an opportunity to view one of those instructional resources. But for a little bit more information about the PGLO plasmid, we have the plasmid map included on the website, as well as links to our instructional videos, the instruction manual, prep manual, student facing manuals. Again, including in some of these lab resources or instructional videos, which might be helpful to you guys within your particular roles.
You've actually got a hand raised, Tamika, so yeah, go across the questions. You can unmute yourself now. I can't read your name, the person the hand raised, Colnaghy. But if you click the mic, you should be able to talk now if you do have a question. Hand's disappeared. Might have raised it by mistake. Sorry to make it carry on.
No worries, no worries, no worries. We also have a YouTube channel which includes it includes a great deal of video resources to help to build the conceptual background for students for this lab, as well as some of our other kits and labs. All right, and then this has given us a fuller look at our social distancing lab adaptations. So again, on the website, we provide just for specific kits, PGLO being one of them, a link to, on the product page, a link to these adaptations, which lays out, again, a summary of modifications, overview of the lab, a visual representation of the student stations, as well as aliquoting modifications, and then any modified, if any, student protocols that lends or that connect with that particular modified protocol or adaptation. So with that, we're at the end of our time together. Thank you for the questions that were presented throughout the webinar, but I will also take any other outstanding questions that you have at this point.
Sorry, I was talking the whole time and I was on unmute myself. But yes, we have a question come in the chat. It's asking if someone's had a PGLO kit for over a year, is it worth running it or should they replace all the reagents?
Define over a year. So how much longer is it now? Company position, so that you're very clear is that we recommend not using it after a year. Sometimes, I mean, it is a good lab that the reagents do sometimes withstand the test of time, but I can't necessarily say I recommend that you do it after you found it after a year. You could try it.